Okay, so it's really totally amazing for me to be here. I had absolutely no idea what I was getting myself into, and it's such a positive surprise for me. I'm really happy to be here, and I think there are many people, excuse me, sitting here who are also very happy to be here. It's a privilege. I was a bit nervous when I saw the program, thinking, after lunch, that's like the killer hour. Everybody's, full from eating and whatnot. So I thought, okay, now we have this technology talk. Great, everybody's just going to stay awake, right? But what was more worrying for me was that normally this topic is not, as Topi mentioned, talking about bugs, some people think it's gross, right? And so having this topic right after lunch, I thought, maybe it'll turn some people off as well, I don't have to worry about my topic anymore because, thank you, Topi, you went first. Okay, so normally I am doing a lot of research, but also a lot of teaching. And what I wanted to do today is to give you a little lecture. I wanted to tell you about cultured meat and how we actually do it, because I think most of you have heard about this before, but you might not really have an idea or a clear picture about how we do it. So I wanted to give you some information about how one might be able to grow meat. And then I want to talk about what's happened since the world's first cultured meat hamburger was made and eaten about 2.5 years ago. And then if we have time, we talk about what might be in the future, okay? And interrupt me whenever you want to ask questions or, yes. So I was prepared to speak in Swedish today, but we switched to English. So all my slides are in Swedish, but I'll be speaking English. Okay. So I think many people are aware of the problems with today's production of meat that's been around the news, the media, everything. A lot of issues regarding ethical aspects, environmental impact, but there's one point that doesn't really get a lot of attention, I think, and that is the issue of not so long in the future, it will not be possible for us to produce enough meat to meet the demand, the projected demand in the future, because of the issue of land area. Okay, we're currently using about 60% of all of the usable land area for on the Earth now for meat production, and that includes feed production as well. And if we're going to double our production of meat in order to meet the projected demand, that's just not going to happen. It's not possible. It's not feasible for us to do that. So we have to come up with some alternatives, and one is, which we go out and we hear a lot in the media, that we have to eat less meat. And then there's the possibility of looking for an alternative for meat. So this idea of what we call cultured meats, some people have heard it as in vitro meat, lab meat, synthetic meat. We have chosen to use the term cultured meat for a couple of years now. There's been a life cycle analysis performed to try to identify what would be the benefits in terms of energy usage, land usage, water, environmental impact, etc., if we were to produce a cultured meat product. And I haven't even told you what it is yet, but you can look at either one of these screens here, and there's a graph there, and I think the only thing that you can see very clearly is the size of the different circles, and the big red one is beef, and we have, there's no pointer here. We have lamb is the pink one, pork, the lighter pink, poultry is yellow, and the little green dot is cultured meat. And the size of this dot, these circles, is giving you an idea of the greenhouse gas emission for these different things. So you can see that based on the calculations for land use as well, there would be definitely a benefit to cultured meat compared to the other products. The only thing which maybe is better, is poultry, and that is that it consumes less energy to produce for the same amount of meat. So some people who are working with this have made these calculations and to argue, yes, there are a lot of benefits for producing cultured meat. The only problem is that cultured meat doesn't really exist. There have been a couple of prototypes, but we really don't know how we're really going to go about making it. So the process to make it isn't defined. And therefore, when you do this life cycle analysis to calculate how much energy and whatnot is used, it's based on a lot of assumptions, and it's based on a process which doesn't exist. So we think it's going to have be better for the environment, et cetera, but the actual, the jury is still out on that one. Okay, so how do we grow meat? And that is what cultured meat is. It's about growing meat outside of the animal's body. And we do that by using methods that we take from a very similar field of research and development, and that is to grow tissues and organs for humans, for transplantation. And this is something which has been around for a long time. It started like 25, 30 years ago. And actually growing meat is just a spin-off of that. And the reason being that meat is one of the animal's body parts. It's a skeletal muscle. So when it comes to the area, what we call tissue engineering, this idea of growing tissues and organs for humans, this is a very interesting picture because it showed up in a journal, a magazine called Scientist about 10 years ago, actually now. It's been the dream for the surgeons and the doctors to have an off-the-shelf product. So a patient comes in, they need a new heart, we go to the shelf, we take down a living heart and we implant it. And the picture they use to show this is a heart wrapped up like something you would buy at the food store. And that is basically the spin-off idea, because to grow meat, which means to grow skeletal muscle, it's actually easier to eat. It's easier than growing skeletal muscle that we would be implanting into a person because it doesn't have to function. It just has to provide nutrition, taste good, and be something desirable to eat, which is not so easy. So what I want to do is take a couple of minutes to give you this little mini lecture about how do we grow tissues and organs so that you have an idea about what we're talking about. And to do this, we basically have 3 or 4 main ingredients or requirements to do that. We need to have living cells. And this is a picture, if you look through a microscope, you'll see living cells. And these cells we usually use are stem cells because they have they can reproduce to large numbers of cells. We need to have something which the cells are going to attach to. And what we're normally using is something what we call a scaffold, or byggnadsställning in Swedish, and it's a temporary structure, always porous. There's a picture here in the middle. If you are sitting close, you'll see it looks like a sponge. But you can have a porous material which is made fibers like a cloth, and the idea is that you have these pores, and the cells like to attach to the material so they can grow and migrate into the center of this construct or material, which you can shape. If you were to be, for example, growing a liver or an ear or a heart, you could shape this scaffold into the shape that you would eventually like to have. But the most important thing about the scaffold is that it needs to be degradable, so that with time it will be disappearing as the cells are starting to build new tissue. And that's what we see here in a picture. There are cells which have been fakely colored red, are growing on these fibers, which are degradable in the scaffold. And they, like these fibers, they grow in through the whole structure. And then what they do is they start producing tissue. And if you can see in this picture, for those of you who are sitting close, the cells have started to make these thin white little fibers. And this is the beginning of the tissue. So with time, these cells are going to start making tissue, as they would normally in the body, and the scaffolding material will be degrading. And at the end of the day or weeks, you will be left with only the pure natural tissue that the cells themselves have made. Now, in order to be able to tell the cells to control what the cells are doing, we have to give them a lot of information. We have to tell these stem cells, okay, we want you to make skeletal muscle, and we want you to become a muscle cell. We don't want you to make bone, for example. So we need to give them different signaling molecules, and these are the normal. We have to learn how the body does it naturally, and then we try to mimic that outside of the body. So these are different growth factors, and different molecules that the cells normally use to talk to each other. So we have to figure that out. And then finally, we need to do all of this in a very sterile environment. We call it a bioreactor. We need to keep the temperature 37 degrees. We control that there's no microorganisms. We be able to feed the cells with the nutrients and things that they need so that they can grow. They can increase in number and form a tissue. So this bioreactor becomes like the animal's body. So what we're doing is we're using, we're mimicking what's going on in the body, but we're doing it outside of the body. Okay, how far have we come? Well, we have living skin. It's been on the market for almost 25 years now. You can buy living human skin to transplant and to use on, for example, burn victims. We have several years ago, 2002, the first urinary bladder was made. We have a picture of one here in a beaker that you would find like in a chemistry lab. And this was made actually for a dog, but now this is a big commercial product in the United States for many men and women. And we also have, now it hasn't been used in humans, but in animals we've been able to regenerate a beating heart. And I'll come back to this hopefully later. But these are all things that have happened for health reasons, on humans, right? So what's the deal with cultured meat? Well, as you all know, this cultured meat is skeletal muscle. So we need to work on tissue engineering skeletal muscle. And if we take a look at what muscle actually is in the microscope, what you see, and many of you know this better than I, probably, you have this fibrous structure. The muscle forms muscle fibers. And if we want to try to grow muscle, we have to make sure that those muscle fibers are oriented parallel to each other so that we can recreate the same structure that a natural, native muscle has. And that, my friends, is one of the trickiest things to do on the one hand. Okay, it's one of the tricks. So what you see here is these are a series of petri dishes, so cell culture dishes, all lined up in a row. And there's a zoom in on one of them here, and there's a thin string in the middle there. And that thin string, as of about 7 years ago, was the largest piece of human muscle that was made in order to, would be put into a human. We wouldn't be eating this, don't get excited. But this was only about 2 centimeters long and less than 1/2 centimeter wide. And that was as large as it could go. Why? There are no blood vessels in this system. And everything, the oxygen and the nutrients that the cells need in order to grow and to live, goes by diffusion. So once the tissue gets too large, the oxygen and the nutrients can't get into the center of the tissue. So nature has created a blood vascular system for us, so we need to be able to make a blood vascular system inside of a growing tissue in order to be able to make something big, like a steak. But we haven't achieved that yet, even in the tissue engineering field, but we're very close to doing that. Okay. How am I doing on time? Okay, good. So when we talk about meat, and again, I feel like I'm preaching to the choir, we talk about 2 different types of cultured meat. The first type would be to be able to grow a whole skeletal muscle, like I just mentioned before, a big piece of muscle. But in order to be able to do that, we need to have at least a blood vessel system, and we haven't quite gotten there yet. But the other thing, which is a lot easier, is to be able to grow chop meat or mince meat, köttfärs, because then we only need to grow the small pieces of muscle and just gather them together. And this is where the field has actually started, and the first successes and the prototypes, the proof of concept have come. So I want to show you 3 approaches that have been used to make cultured meat. We have a cartoon here. I call it, it's called Chop Meat 1. And this is a cartoon that was published in the journal Nature some years ago, about 6 years ago. And the idea is this, that you actually, you have an animal. It's a pig, a cow, what have you. And you go in and you take a biopsy. So it's a small tissue sample, and there are special biopsy punches. Some of you might have had some spots on your skin biopsied. You take a bit of the animal's muscle, and you take it out into the culture dish, and you extract out what we call the muscle progenitors, or the stem cells that will eventually become muscle. And you let them grow in the lab, and you increase in number, increase in number, increase in number. And that's what stem cells are really good to do. They can make lots of cells, because if you're going to be growing, if you want to have lots of meat, you need lots of cells. So you have to give them different signaling molecules. And then once you get to the right number, then you have to tell these cells, okay, I want you to be a muscle cell. So they need to switch and to start to mature into a muscle cell. And when it starts to do that, they line up to each other, they start to fuse, they form things called muscle myotubes, and eventually the beginning of fibers. Now, we all know that if we go to the gym, we get bigger muscles, right? And that actually is the case even in the Petri dish in the lab. If you stimulate either mechanically or electrically muscle cells and these muscle fibers, they will actually increase in size, and you will get more protein production. So a lot of researchers have been trying to mechanically stimulate, pulling on the cells or electrically stimulating them in order to get more protein and bigger muscle fibers. But at the end of the day, you would collect all of these fibers together. And the cartoon here is showing you add different flavoring agents, maybe some iron, fat, because all we have is muscle cell. We don't have fat now. We mix it all together, and out comes the sausage at the other end of the cartoon here, and you could cook it and eat it. And this was, in principle, the approach that Mark Post used to make the first hamburger 2.5 years ago. Mark Post is also a researcher in the same field as I, down in Holland. And they started off using this approach, all these different Petri dishes, having these little pieces of a muscle growing between 2 silk pads, and they would go in and individually pick each one up, hold on to the ends, and mechanically stretch it, put it to the gym, put it back, go back the next day. And you might have heard that this hamburger costs like 200 or 300,000 euro to make. It's mainly the labor of all the people who were working in the lab to grow all these pieces. They would put them in the freezer, and then at the end of the 3 years, they took it all out, and then they put it together with some binder material. But anyway, that's a very labor-intensive way to go about doing it. And here's a picture of from the PR event 2.5 years ago for the first hamburger, and it was being tasted. There's a slight, the process that he actually wound up using at the end of the day was a bit different. I'm not going to show you because we don't have time, but if any of you are interested, I'm going to refer you to a website of an organization called New Harvest, and there you'll find links to, they have videos about how they make it and everything. Okay, mincemeat 2, I'll go quickly now. That is something which you all have heard about, and that's 3D printing. And the idea is now you 3D print cells that are in gels, like an alginate gel, and you print them down out of a printer head, and you build up your skeletal muscle to the point where you get something, you let it grow, and then you have something that you can eat. And here is a picture of Gabor Forax, who is started a company called Modern Meadow in the States, and he is giving a TED Talk here, and this was actually before Mark Post's hamburger, and this white, slimy, slug-looking thing, this was the meat that he grew, and he cooked it in a little steak frying pan and ate it on stage, and he said it didn't taste like anything, because it's just muscle cells, there is no taste, which can make you wonder, why do we actually need to use muscle cells? The third approach is one of the approaches that we've been doing in Gothenburg, but other people are also using it, and I think this is the technique behind the latest company that started up called Memphis Meats, and just a month or so ago they showed the production of a meatball, which created a lot of attention. But in this case, you're growing the cells on the surface of these small microcarrier beads, and they're small spheres. They're just some less than a millimeter in size, and the cells grow all over the surface, so you're making a large surface area, lots of beads floating around. And these microspheres, in our case, we're working with a Swedish company, a starch company, so these are made out of starch. So you allow the muscle cells, which are shown here, to grow on these starch microspheres, which are shown in the next picture. They cover the entire microsphere, and then they start growing off of the microsphere, and they start building muscle fibers and connecting to the different spheres. And at the end of the day, you get this big gegamoya, which you can retract. But now you can eat the scaffold in this case. It's starch. You don't have to worry about it degrading. And all this is done in a bioreactor. And here is a picture for those of you who are sitting close. You can see a close-up of this bioreactor, and all the little red dots there, these are these microcarrier beads coated in the muscle cells. Okay, that's the end of my lesson. What happens now? Now we have this New Harvest organization. I see I spelled Isha's last name, Isha Datar. There's an R at the end. They have now expanded beyond just making meat without animals is the catchphrase. They have been supporting new startups like Moo Free, which is making milk by recombinant techniques. So you get the bacteria and yeast to make all of the proteins that you would find normally in milk, and you mix them together. Clara Foods making eggs without chickens, and again, making bacteria, so you put a gene for the proteins that you would find in egg, like albumin, and you get the bacteria to make those proteins instead, and you extract them, and then you make egg white, the white part of the egg out of protein. There's a general term now, which is instead of cultured meat, there's a bigger concept now, or större begrepp, cellular agriculture. And this is the latest term to come out to describe all of this.